What is evolution? Evolution is the development of life on Earth, a continuous process that began billions of years ago and persists to this day. Evolution explains how the enormous diversity of life was able to develop. It demonstrates how primitive protozoans could diversify into the millions of distinct species we observe today. Evolution, then, is the answer to a question that many of us have asked when seeing a Dachshund and a Great Dane together. How is it possible for common ancestors to have descendants that look so remarkably different from them? In answering this question, this script focuses specifically on animals, excluding other kingdoms of life such as fungi and plants. The fundamental question to ask is therefore, how can one animal lineage diverge and develop into an entirely new species? But first, a quick question: what exactly is a species? A species is a community of organisms capable of interbreeding to produce fertile offspring, meaning their offspring are also capable of reproducing in turn. To understand this mechanism thoroughly, we need to take a closer look at three core pillars: individual uniqueness, guaranteed through the overproduction of offspring and heredity; genetic variations, recombination and mutation; and natural selection. Let's begin with uniqueness. Every organism is unique, and this variation is essential for evolution. While members of a species may strongly resemble each other in appearance, they all possess slightly different traits and characteristics. They may be a bit larger, heavier, stronger, or bolder than their peers. What drives these individual differences? Let's look closer at the cellular level. Every organism is composed of cells, each containing a nucleus. The nucleus houses the chromosomes, which carry the DNA. DNA consists of various genes, the fundamental information carriers of life. These genes contain instructions for the cells, determining the structural and behavioral traits that living creatures exhibit. Because each individual's DNA is unique, it introduces slight variations from one individual to another, resulting in distinct characteristics. But how is this vast array of unique DNA generated? One key ecological factor is the overproduction of offspring. In nature, organisms generally produce far more offspring than are necessary for the baseline survival of their species. Consequently, many offspring experience early mortality. Often, population sizes exceed the carrying capacity of their environment. This overproduction acts as a catalyst for diversity. The more offspring produced, the higher the statistical probability that minor genetic differences will emerge. This is precisely how nature operates, maximizing variations within a population. The second major driver of individual uniqueness lies within the process of heredity itself, the transmission of DNA from parents to offspring. Two critical mechanisms drive this process: recombination and mutation. Recombination is the random mixing of genetic material from two organisms. When two animals mate, their genes are recombined twice. The first instance occurs independently during gametogenesis, the formation of gametes, sperm and egg cells. During this stage, gametes receive a randomized half of the parental chromosomes. The second recombination occurs during fertilization. Each parent contributes exactly 50% of their DNA, blending their unique traits. The resulting offspring inherit a completely novel, randomized combination of parental DNA, significantly accelerating phenotypic diversity. Conversely, mutations are spontaneous, permanent alterations in the DNA sequence. These can be conceptualized as replication errors within the DNA, triggered by environmental factors like toxins, chemical substances, or ionizing radiation. A mutation occurs when a segment of the nucleotide sequence is permanently altered. While many mutations are deleterious or neutral, sometimes leading to pathologies like cancer, some yield beneficial or benign traits. For example, the emergence of blue eyes in humans originated from a single, random genetic mutation. Crucially, for a mutation to influence evolution, it must occur within a germline cell, a sperm or egg cell, as only germline DNA is inheritable. This explains why medical professionals shield reproductive organs during X-ray procedures, while somatic tissues face lower long-term evolutionary risks. In summary, during heredity, Organisms transmit their traits via DNA. Recombination and mutation alter this genetic blueprint so that siblings remain distinct from one another, each inheriting a unique configuration of parental traits. The definitive word here is random. These foundational mechanisms rely entirely on chance. If these underlying mechanisms are driven entirely by chance, how do living organisms become so exquisitely adapted to their environments, such as the cryptic stick insect, the specialized hummingbird, or the well-camouflaged frogfish? The answer lies in the second core pillar, selection. Every individual is subjected to natural selection. Because extensive variation exists within a species, environmental pressures act as filters. These selection pressures include predators, parasites, intra-species competition, toxins, habitat shifts, and climatic changes. Every creature possesses a unique combination of traits that either enhances or hinders its survival metrics. Individuals with maladaptive traits are selected against by their environment, often dying before reproducing. Conversely, those possessing adaptive traits survive to reproductive age, passing those advantageous genes to the next generation. This underlines why biodiversity is vital. By generating diverse offspring, a species maximizes the mathematical probability that at least some individuals will survive environmental selection filters, ensuring lineage persistence. A classic empirical demonstration of this process is observed in the finch populations of the Galapagos Islands, famously documented by Charles Darwin. Centuries ago, a small group of ancestral finches was displaced to the isolated archipelago, likely driven by a severe storm. They arrived in a pristine environment with abundant resources and an absence of natural predators. The population expanded exponentially, quickly reaching the island's carrying capacity. As resources grew scarce, intense intra-species competition triggered strong selective pressures. Minor variations in beak morphology, which initially seemed trivial, suddenly became defining survival assets. Finches with stouter beaks excelled at cracking seeds, while those with slender beaks were better suited for extracting insects and worms. Consequently, different groups sought out distinct ecological niches to mitigate competition. Over generations, assortative mating within these niches reinforced these specialized traits. Eventually, the morphological and genetic divergence between seed crackers and insect eaters grew so pronounced that they could no longer interbreed. Speciation had occurred. Today, 14 distinct finch species inhabit the Galapagos, all radiating from a single ancestral group. This is how new species originate, through the continuous interplay of individual uniqueness, overproduction of offspring, genetic recombination, mutation, and natural selection. Understanding this framework clarifies the origin of biodiversity and the precise adaptation of organisms to their habitats. Ultimately, this process shapes our own existence. Every human being represents the culmination of 3.5 billion years of unbroken evolutionary adaptation. Given that over 99% of all species that have ever existed are now extinct, your presence today is a profound biological success story. The dinosaurs have vanished, yet you are here, a unique, irreproducible product of the universe.